So this is the overview of male reproductive organ, where our syllabus will focus on testes. Testes is plural, while testis is singular. So inside the testis is where the process of spermatogenesis will occur. Once the sperm is produced inside the testis, the sperm will be stored inside the epididymis, near the testis. This is the structure of sperm, where its function is to carry father's genetic material to the egg. So let's go deeper. The specific structure and function of sperm. But before that, let's discuss about the specific location of spermatogenesis. So inside the testis is a long and coiled tubule known as seminiferous tubule. So inside the seminiferous tubule is the specific location of spermatogenesis. If in exam, the question asks, where is the specific location of spermatogenesis? You must mention seminiferous tubule of testis. And then once the sperm is produced inside the seminiferous tubule, the sperms will swim and stored inside the epididymis, another long and coiled tubule but located outside the testis, known as epididymis. This is where the sperms will be stored. So let's discuss about the function of specific structure in sperm. Sperms divides into three main parts: the head, middle piece, and tail. Head and middle piece is connected by neck. So inside the head are acrosome and nucleus. Inside the middle piece is a spiral shape of mitochondrion. The tail is made up by a flagella. So what is the function of acrosome inside the head? Acrosome contain hydrolytic or digestive enzyme which will digest the zona pellucida of egg. Zona pellucida is a layer that protects the egg before the nucleus of sperm fuse with the nucleus of egg. The sperm must penetrate the zona pellucida. So the acrosome will help the sperm to penetrate the zona pellucida. What is the function of nucleus inside the head? The nucleus carry the father's genetic material. What is the function of mitochondrion inside the middle piece? Mitochondrion will provide energy for the sperms to swim and fertilize the egg. And the last part is tail. Its function is to help the sperms to move and swim towards the egg. In spermatogenesis process or in production of sperm process, there are many stages of cell occur before a sperm is produced. As we had learned just now, the specific location of spermatogenesis is occur inside the seminiferous tubule of testis. So, this is a cross-section of seminiferous tubule. This is the wall of seminiferous tubule, and this is the lumen. At the center of the tubule is a lumen. The spermatogenesis occur in the wall of the seminiferous tubule. So, from the basal lamina towards the lumen, that is the direction of the spermatogenesis process. So, firstly, the spermatogenesis is started inside the seminiferous tubule of testis. Start from embryo stage. In the testis during embryo stage, the primordial germ cells will undergo differentiation to form spermatogonium, which is also diploid. All the primordial germ cells must undergo differentiation into spermatogonium during embryo into fetus stage, so that after birth, the baby boy will already has a stock of spermatogonium inside the seminiferous tubule of testis. So once the boy reach puberty, the spermatogonium will undergo mitosis to produce two spermatogonium cell. One of the spermatogonium will remain as spermatogonium, while the other one will undergo differentiation, forming a primary spermatocyte, which is also diploid. So what happened to the primary spermatocyte? It will undergo meiosis 1 to produce two haploid secondary spermatocyte. And then each of the secondary spermatocyte undergoes meiosis 2 to produce two spermatid. From one primary spermatocyte, there are 4 spermatids produced. So what happened to each of these haploid spermatid? They must undergo a process known as spermiogenesis before each spermatid develops into sperms with specific structure. So what is spermiogenesis process? Spermiogenesis is a process where the spermatid undergoes differentiation into a specific structure. This process requires nutrients, where the nutrients will be supplied by Sertoli cells. Sertoli cells is a cell found inside the seminiferous tubule. It facilitates the process of spermatogenesis. So, let's look at this diagram. We can see that from spermatogonium, primary spermatocyte is produced, and then spermatids are produced, and then the spermatozoa will be produced. The direction is from the basal lamina towards the lumen of seminiferous tubule. So, what happened to the spermatozoa? The spermatozoa will be released into the lumen, and then it will swim towards the epididymis to be stored. So, let's discuss why some of the spermatogonium will remain as spermatogonium and not continue undergoes differentiation forming primary spermatocyte. What is the important to maintain the existence of spermatogonium in the seminiferous tubule? This will ensure the continuity of sperm's production in a long run in the future. So means that as long as there are spermatogonium inside the seminiferous tubule, the spermatogenesis process will always occur. Hence, sperms will always be produced. That's why men will always produce sperms in their whole life until they die. There are 5 types of hormone involved in spermatogenesis. Gonadotropin releasing hormone, GnRH. GRH is in capital letter, N must be written in small letter. Luteinizing hormone, LH, both is in capital letter. Follicle stimulating hormone, or FSH, in capital letter. Testosterone and inhibin. So, which cell or structure secrete all of these hormones is required in our syllabus other than function of each hormone. So, when the boy reach puberty, the boy will started to secrete gonadotropin releasing hormone. So, which part of the body secrete gonadotropin releasing hormone is the hypothalamus. Hypothalamus is located at the brain. So, hypothalamus secrete gonadotropin releasing hormone to stimulate the anterior pituitary gland. Anterior pituitary gland located also in the brain near the hypothalamus. So, GnRH stimulate anterior pituitary gland to secrete FSH and also LH. FSH and LH will be secreted into the blood and transported inside the blood towards testes. Inside the testes, there are 2 types of cells affected by each of these hormone. The 2 cells are Leydig cells and also the Sertoli cells. So, FSH will stimulate Sertoli cells to promote spermatogenesis. At the same time, LH will stimulate the Leydig cells to secrete testosterone, which will facilitate the spermatogenesis. So, testosterone secreted by the Leydig cells will facilitate the process of spermatogenesis and also promote the development of male secondary sexual characteristic, where testosterone will increase the growth hormone inside men. Hence, once the boy reach puberty, the boy will started to develop more muscles and becomes more hairy. So, this is the positive feedback mechanisms of hormones in spermatogenesis. Positive feedback mechanisms means when one process occurs, it will enhance another process. The easiest way to remember which hormone stimulate which cell is FSH has S. Between Sertoli cell and Leydig cell, Sertoli started with S. So FSH stimulate Sertoli cells. LH has L and Leydig cell started with L. So LH will stimulate Leydig cells. Other than positive feedback mechanisms of hormones in spermatogenesis, there is a negative feedback as well. Negative feedback mechanisms means when when one process inhibits another process. So the first negative feedback is when the level of FSH is higher than normal. So if the FSH level is higher than normal, this will stimulate Sertoli cells to secrete inhibin. Inhibin will give negative feedback to the anterior pituitary, where it will inhibit the anterior pituitary gland from secreting FSH and LH. Hence, level of FSH and LH will be decreased. When the FSH level decrease, this will return the level of FSH back to normal. So this is the first negative feedback mechanism. The second negative feedback mechanism in spermatogenesis occur when the testosterone level is higher than normal, which causing actively production of sperms. Actively production of sperm will causing high sperms count, means that there are too much production of sperms. This situation will causing the testosterone itself to give negative feedback to hypothalamus and anterior pituitary gland, where testosterone inhibit hypothalamus from secreting GnRH and also inhibit anterior pituitary gland from secreting FSH and LH, causing GnRH level will decrease and the FSH and LH level also will be decreased. Decreasing in the LH level will causing Leydig cells to decrease secretion of testosterone. Hence, the testosterone level will be returned to normal and sperm count will also return to normal. As a conclusion, in spermatogenesis, there are 5 types of hormones involved. Each hormone has specific function and is secreted by specific structure. The first hormone is gonadotropin-releasing hormone, which is secreted by hypothalamus. Its function is to stimulate anterior pituitary gland. The second hormone is luteinizing hormone, which is secreted by anterior pituitary gland. Its function is to stimulate Leydig cell in testes. The third hormone is follicle-stimulating hormone, secreted by anterior pituitary gland. Its function is to stimulate Sertoli cells in testes. Another hormone is testosterone, secreted by Leydig cell in testes, where its function is to facilitate spermatogenesis and promote the male secondary sexual characteristic, and also it can inhibit hypothalamus and anterior pituitary gland if the testosterone level and the sperm's count is higher than normal. And the last hormone is inhibin. Inhibin is secreted by Sertoli cells in testes only if level of FSH is higher than normal. So inhibin only synthesized if the level of FSH is too high. The function of inhibin is to inhibit the anterior pituitary gland from secreting FSH so that the level of FSH will decrease and return to normal.